We will finish up the digestive tract by talking about the small and large intestine. All of this is going to be histology focused. But before that, let's talk a little bit about the small intestine. So, fun fact, this guy is about 17 feet long in living humans, and it is referred to based on its diameter, not the length. So the diameter of the small intestine is about 1.5 centimeters when it is empty. That's why it is considered smaller in diameter than our large intestine. But lengthwise, I know a 2-story house is 20 to 25 feet in height. So your small intestine stretched out is almost the height of a 2-story house, for better or for worse. So small intestine is mainly responsible for absorbing nutrients, and the histology does reflect this. So we have throughout the small intestine numerous folds into the lumen. And we can see that pattern really nicely on the slide. I just pulled this from Google, and you can do the same as you're studying. So these are circular folds called plicae circularis. That is Plicae circularis. And all these do is help slow down the passage of material through the small intestine, as well as increase surface area for absorption. So along all of these folds, we are going to see villi. Those villi are again helping to absorb nutrients. So the presence of these folds and of the villi and the microvilli, all of those will maximize surface area within the small intestine for absorption of nutrients. There is a helpful diagram. So if you ever get caught up in this kind of microscope perspective and you want to see a better diagram, I think this diagram does a good job of showing how we've got the small intestine, which is just one part of that digestive tract, that gut tube. And then if we zoom in, we will see these really circular projections into that lumen. And lining each of these, we have finger-like projections called villi. So this big unit is going to be our plicae circularis. And then along each of those units, we have villi. But we still have the same layers that we saw in the rest of the digestive system. We still got mucosa, in this case with the plicae circularis. We'll still have a lighter layer of submucosa and our muscularis externa layer, and then either a serosa or an adventitia. In this part of the digestive tract, we'll mostly be dealing with serosa. All right, let's jump into our histology. So the small intestine is divided into 3 sections. While you do not need to be able to identify the separate sections on a macroscopic model, such as our muscle man, you do need to know these 3 distinct regions under a microscope. So that is a lot of what we will focus on today. So this is a slide of small intestine. For whatever reason, the Human Protein Atlas divides the small intestine into the duodenum, and then the other 2 are on a separate slide. So we'll just look at those. The way our microscopes work in class is we have one slide with 3 separate dots, and each of those dots is a different part of our small intestine. And so what you will need to know for the exam is how to identify the 3 distinct regions from that slide. So that is something that we will practice in class on December 1st. But for now, let's go ahead and take a look at our duodenum, which is that first part of the small intestine. So this is that part that is coming from the stomach, and it is just a little C-shaped loop that is going to become the middle portion, the jejunum. Histologically, we are going to see lots and lots of villi. And while we will find villi throughout the entire small intestine, they are a little more frequent in the first 2 parts of the small intestine, because that is where most of our absorption of nutrients is going to happen. As we move further along the tube, the waste product will get pushed towards that terminal part of the small intestine before it enters the colon. We will see the same pattern within the mucosa, where we have goblet cells, which are these guys in white that you can see here, kind of scattered all along the surface of those plicae circularis. On our plicae, we've got these additional folds called villi. Let's see which, okay, so depending on where you're at, these villi can be easier to see. we've got a really nice section right here. So that is what our human protein atlas is directing us to. So within each of those plicae circularis, we're going to have these finger-like projections called villi, and these are really nice. They're nice and long. We can see that they are projecting into the lumen and helping to maximize that surface area for absorption. If we move a little deeper now, we get into the submucosa. And this submucosa is very busy. We have these additional glands called duodenal glands, or sometimes you'll see the term Brunner's glands. In this case, the Human Protein Atlas calls them Brunner's glands. But these guys are going to secrete mucin, and their main function is to protect the small intestine tissue from the acidic contents. So we have to remember the duodenum is receiving chyme from our stomach. At this point, the chyme has been exposed to pepsinogen, hydrochloric acid, and gastric juices. So it is usually pretty highly acidic in content. And so these Brunner's glands are secreting mucin to help protect the small intestine tissue from those acidic substances. And that is going to be the main distinguishing feature of our duodenum. Let's move on to jejunum now. Jejunum and ileum are going to be found on this same slide. So how the slide works is towards the left we have our jejunum, and it is eventually going to transition into our ileum. So this is actually a great way to practice identifying the regions because we don't have them nicely separated for us. The jejunum is our middle portion of the small intestine, and it's actually really generic. There are no special features within the jejunum that are going to help us to identify it, and so we need to use process of elimination to figure out what it is. So if we zoom in here, we have our mucosa in this darker purple. We're going to have our underlying submucosa, and you can see we don't have those Brunner's glands or duodenal glands anymore. They have gone away. So the main part of this jejunum is it's still absorbing nutrients, but at this point the acid from our stomach has been neutralized, so we don't require those Brunner's glands to help protect the small intestine. So those have gone away. We still have this pattern here with plicae circularis and all of these finger-like villi coming off. But beyond that, it is a pretty generic material. Let's go ahead and move on to our ileum. So we're going to move down the tube here. And in the ileum, we are looking for actually a lymphatic structure. And I did bring this up briefly during our lymphatic and respiratory lab, but the ileum has a cluster of lymphatic nodules, and in this case we call them Peyer's patches. So what we're going to look for on this slide is these darker circular patches, very similar to those lymphatic nodules we saw within the lymph node, and they're going to look a little like this. So it's just a slight change in texture and color from the surrounding mucosa. But this is going to be one Peyer's patch, and I think it helps a little to zoom out. So you can see that circular shape from afar, but when we zoom in, you can see this is just a little patch of lymphatic tissue. Generally, we're going to find Peyer's patches within the mucosa, but this is not a set rule. Sometimes you'll see them in between the mucosa and the submucosa, but we want to look at that border to find these Peyer's patches. So to review, small intestine has 3 regions. The first and the last region have unique features, but the middle region does not. So when you're looking on your slide, how you know you're dealing with duodenum is if you see this really busy submucosa with many, many Brunner's glands. I'm going to skip over jejunum for now. If you see the presence of those little patches, those Peyer's patches, that's how you know you're dealing with the ileum. And really all these are doing is just monitoring pathogens within the small intestine, making sure everything is getting neutralized, part of that immune function before we send that material to the colon. And then in between we have this generic region called the jejunum. So by process of elimination, if we don't see Peyer's patches within the submucosa, and if we don't see any of those Brunner's glands, that's how we know we're dealing with the jejunum. Last section to talk about is the colon. All right, so we did gloss over some structures such as the cecum. The cecum is a little pouch that connects the ileum and the colon. So we're not going to see that histologically. It is also an attachment point for our appendix, which we also don't spend a lot of time on. If you would like to know further about those, I'm happy to talk about them in class. But for now, we're just going to focus on the actual colon. So in addition to those macroscopic structures, you need to know things like ascending, descending, and transverse colon, as well as the flexures. You also need to be able to identify it under a microscope. And right away, you can see this does look a little different than our small intestine. So let me flip back for a minute. This is our small intestine. Note color and texture. If we switch back to our colon, it's a little lighter in color. And we also have a very, very busy mucosa. So the main histological feature of the colon is the presence of tons and tons of goblet cells. Like, I'm talking most of the mucosa here is taken up by goblet cells. What the Human Protein Atlas is directing us to is just a specific section here. This is what you'll see when you first put your slide under a microscope, and then we have to zoom in a little bit in order to see those goblet cells. But once you do, they are going to be just everywhere within that mucosa. And that is the main feature. We still have, again, our 4 main layers of tissue, but in this case, we're just mostly focused on the mucosa. So under the digestive tract module, we have this slide called colon, tinea coli, monkey. And what we see here, same pattern. We've got our lumen. The next layer is going to be our mucosa. We'll have our underlying submucosa of connective tissue. And then we get into that muscularis external layer. And this is where we will find our tinea coli. In this case, the tinea coli will show up like this little bump. It's just like an extra ridge of muscle. And if you can imagine that running along the entire length of the colon, because it is a little bit shorter than the actual length of the colon, it's going to cause that bunching up appearance. And that's how we get these haustrum, these pouches that are along the surface of the colon. We can also see those haustrum and the tinea coli from a macroscopic view on our model, so make sure you're comfortable with the concept from both approaches. Lastly, we do have these little fatty tags, and those don't really show up well in slides, so let's see if we can't find an example. So once again, Google is our friend. I did do a little bit of searching. I was trying to find a cadaveric photo to show you guys, but this one will do. So we have these epiploic appendages, that is EPI, PLOIC, appendage. And all these do, all they are is little fatty appendages lining the colon. So that is another way that you can identify it that makes it distinct from the other regions. As far as what they do, we have no idea. So that is an ongoing area of research. We don't know what they do, but we do know they are associated with the colon, mostly within the transverse and sigmoid regions. So, in summary, we have covered small intestine and large intestine, or colon. You need to be able to identify the 3 distinct regions of the small intestine under a microscope. Duodenum versus jejunum versus ileum. And you also need to be able to identify the colon. So this guy here with the repeating pattern of goblet cells throughout that mucosa. And that's going to be the main defining feature here. So I will be fair to you guys. I will find a slide that has these numerous goblet cells within its mucosa. All the things we talked about today, all of those unique features, those are need to know information to identify these structures.